Bacterial leaf blight. Xanthomonas oryzae PV. Oryzae. Economic importance. The disease was first observed in Japan 1884. In Indonesia, Kresek disease was reported to kill young seedlings completely in 1950. In India, BLB was first reported in 1959. A severe outbreak of the disease occurred in Bihar and Uttar Pradesh in 1963. In the tropics, the disease is usually referred as bacterial blight as it often kills entire young seedlings. Yield losses in severely diseased fields range from 20 to 30% and occasionally 50%. In India, millions of hectares are infected every year. Yield losses have been as high as 60% in some states and Godavari district of Andhra Pradesh which are endemic to this disease. Taichung native 1 is highly susceptible. Symptoms: the bacterium induces either wilting of plants or leaf blight. Wilt syndrome known as Kresek is seen in seedlings within 3 to 4 weeks after transplanting of the crop. Kresek results either in the death of whole plant or wilting of only a few leaves. The bacterium enters through the hydatodes and cut wounds in the leaf tips, becomes systemic and cause death of entire seedling. The disease is usually noticed at the time of heading but in severe cases occur earlier also. In grown-up plants, water-soaked, translucent lesions appear usually near the leaf margin. The lesions enlarge both in length and width with a wavy margin and turn straw yellow within a few days, covering the entire leaf. As the disease progresses, the lesions cover the entire leaf blade which may turn white or straw-colored. Lesions may also be seen on leaf sheaths in susceptible varieties. Milky or opaque dew drops containing bacterial masses are formed on young lesions in the early morning. They dry up on the surface leaving a white encrustation. The affected grains have discolored spots surrounded by water-soaked areas. If the cut end of leaf is dipped in water, bacterial ooze makes the water turbid. Etiology: the bacterium is strict aerobe, gram-negative, non-spore-forming, rod-shaped with monotrichous polar flagellum off at one end. The bacterial cells are capsulated and are joined to form an aggregate mass. Colonies are circular, convex with entire margins, whitish yellow to straw yellow and opaque. The bacterium has many strains that differ in ability to infect rice plants. Strains in tropical countries are usually more virulent than those in temperate areas like Japan. Bara. Disease cycle: the bacterium enters the plant through water pores, hydatodes along the edges of the leaf and through injuries in roots or leaves. It does not enter through stomata. BLB is primarily avascular or systemic disease. Bacterial cells move along the vascular tissues causing wilting. Rainstorms and typhoons help in the spread of the disease. Irrigation water also carries the organism from field to field. The primary source of infection is through bacterium overwintering in seed, husk and endosperm. Bacteria may survive in soil, plant stubbles and debris. The pathogen also survives on collateral hosts like Leishmania hexandra, Leishmania oryzoides, Zizania latifolia, Cyprus rotundus, Cyprus deformis, Phalaris arundinacea, Cyanodon dactylon, etc. The bacterial ooze serves as secondary inoculum and cause secondary infection. Favorable conditions: clipping of tip of the seedling at the time of transplanting, heavy rain, heavy dew, flooding, deep irrigation water, severe wind, temperature of 25 to 300 C and application of excessive nitrogen, especially late top dressing. Management: 3/4 grow resistant cultivars like MTU 9992, Swarna, Ajay, R20, R42, R50, R54, TKM6, Mashuri, IET 4141, IET 1444, IET 2508, Chinsura Boro, etc. 3/4 resistant donors, Teti, Tadukan, Zenith etc. 3/4 affected stubbles are to be destroyed by burning or through plowing. 3/4 judicious use of nitrogenous fertilizer. 3/4 avoid clipping of tip of seedling at the time of transplanting. 3/4 avoid flooded conditions or drying of the field, not at the time of flowering. 3/4 avoid flow of irrigation water from infected to healthy field. 13. 3/4 remove and destroy weed hosts. 3/4 soaking seeds for 8 hours in agromycin 0.025%, followed by hot water treatment for 10 minutes at 5 to 5.4 OC eradicates the bacterium in the seed. 3/4 spray streptocycline 250 ppm along with copper oxychloride 0.3%.